See the screen.
Henny, sorry.
You all right?
No. Can you move further?
You can get
Thank
the
you.
screen right there. Can't you?
Yeah, thank you.
Okay, so thanks for the invitation to talk. What I'm going to do is tell you about the research that's been going on in my lab for about the last 8 years, mainly into SCAR3. And I'll sort of try and describe what we've been doing. But there's always the person who stands up here doesn't actually do the work. It's those people at the back, 5 of them there, who do most of the work that I do, so that I talk about. So if you've got any questions, just turn around and ask them. I can almost guarantee I won't be able to answer it. So what we do is we try and, I should say this first up, I'm not a clinician, all right? I'm a biochemist, and what my lab tries to do is to understand the proteins that are at the heart of this disease. We try and understand at the molecular level what's going on. And so what I'm going to talk about today is how one protein, in particular SCAR3, how it aggregates and how it potentially causes disease, and how we're thinking about how trying to stop that process. Let's see if this works. All right, so I don't know if you can see that, maybe the lights are a bit too bright, but what my lab studies are proteins, all right, and here's just a collection of pictures of any old protein. And what I want you to get from this slide is that proteins have specific shapes, all right, and all proteins can be sort of, yeah, that's better. All right, all proteins have either what we call helices or sheets, and that's how they fold up. All right, now in the human body there are over 30,000 different proteins, and each one of those has a very unique shape. And if the protein doesn't adopt that shape, it doesn't work. So most of you will be sitting there now, your dinner will be in your stomach, and it will be digested. All right, and in your stomach there are a variety of proteins that are chewing up that food. And if those proteins aren't correctly folded, that won't happen, you won't get the energy you need, you'll become sick. All right, so proteins have to fold, they have to adopt the right shape for them to function. If they don't, you get dysfunction, potentially you get disease. So what we study is how proteins actually fold up. All right, now down here we've just got 3 old proteins, and it doesn't matter what they are, we'll get to the interesting proteins in a few moments. All right, what I want you to get from this is when a protein folds up, it starts from a species that looks like this. All right, this is what we call a polypeptide chain. You can just think of it as a piece of string. All right, now inside your cell, this is what comes off or is produced, and it's this thing that has to fold up. And you can see it's actually quite complicated because it has to go from this piece of string to these sorts of shapes down here. All right, inside the cell, this happens in less than a second. All right, if you ask one of my students to be able to draw one of these shapes, it would probably take about 4 or 5 hours and a few bowls of wine, and they might just be able to draw it. All right, it's an incredibly complicated reaction. All right, but the cell can do this really, really quickly. And what determines how this thing folds up are these things on the side here. These are called side chains. My students at the back should be able to tell you which each one of these is. They're now looking embarrassed because I think I'm going to turn around and ask them what ones these are. All I want you to know is that it's the different side chains on here that tell the protein how to fold up. So if you change any of these side chains, the protein will adopt a different fold. Okay? So the information in here dictates the structures down here. Make sense? Now, I should point out that these structures are simplified. All right? What I'm showing you here is just these, what we call helices and sheets. This is what it actually looks like. All right? It's a lot more complicated. And this is what happens when these side chains come together. But for the sake of today, what I want you to remember is these are the sorts of shapes we're trying to form. Okay, now there's one more level of complexity. All right, so what I've shown you is a polypeptide chain, that string, that has to fold up to the correct shape. What decides that string? What's important in deciding that sequence? And that's down to the DNA. All right? Whatever's in your DNA dictates that string of amino acids there. All right? So as I said, want to come in?
Yeah.
Wait.
Excuse us.
Hello. Can you pass?
Yeah, I'll try. Thank you. My God, no.
Move your chair. Hillary, maybe if you can just move out.
100,000 different proteins, and everything in your body is controlled by proteins. Right? They're the machines that do all of the jobs. So they determine whether you've got straight hair, curly hair, the colour of your eyes. They determine everything. So every biological function is controlled by a protein or a family of proteins. All right, so they're the critical thing. If they don't work, Spanish.
Can I have that book, please? Thank you.
Yeah, you're
Thanks.
Okay, so what I want you to get from this is that the sequence of that bit of protein there, that polypeptide chain, which isn't folded up, right, is dictated by the DNA. All right, I'm not going to go through how they're linked, but what I want you to know is that it's the sequence in the DNA which dictates this polypeptide chain, and then what we study is the next step, is how that chain folds up to give you the protein. All right, but what you can see from this is there's a sort of passage of information. We go from here to here to here. If there are mistakes at any step in that chain, this thing breaks down and you end up with proteins that aren't functional. All right? Likewise, if we change the DNA sequence, we change this sequence, we'll change the way this thing folds up. If we change the DNA sequence, we can change the sequence of the protein that has to fold up. And in the case of disease and misfolding, which is what we're really going to be talking about today, you can form fibrils or aggregates. All right? So most proteins are monomeric, they wander around in solution, they're completely soluble. When this process of folding goes wrong, because the sequence here is different, all right, it gives rise to these fibrils. And these things are the things that deposit within tissues throughout the body and can cause disease. Okay? So what I want you to get from this is you change the DNA sequence in the wrong way, changes this sequence, and then you get misfolding. Now, most of the time, the body can cope with this. It has mechanisms which can repair this. It has mechanisms which can detect that this is wrong and can actually get rid of it. But sometimes it escapes that machinery, and that's when you get these things forming, and that's when we get disease. So what we're studying is this process here, with an aim to be able to try and stop it. Right, does that make sense?
Yeah.
Clear enough? All right, this is something, I was trying to explain this process to my son, and the easiest way to describe it, just to make sure you understand this link between DNA sequence and protein, we sort of talked about how the DNA acts as a map. All right, it acts as a template, and whatever the DNA tells you, you're going to get out at the end. So in terms of a map, you might be driving.
Excuse me, sorry to interrupt again. Can we just make the room to
Yeah.
这也挺难没看到。
Sorry about that.
All right, that's all right.
There we go. There we go.
Thanks,
All right. Thank you.
Don't worry. All right.
Sorry, sorry. Yeah.
I might go down back there.